Hello, everyone. Thank you for joining us for today's webinar, KASP Technology Accelerates Rice Improvement in South Asia. My name is Beth Fry, and I'm a product manager for KASP Technologies at LGC Biosearch Technologies. And I'd like to welcome you all today. And before I introduce today's speaker, we just have a couple of housekeeping notes that you can see on your screen. First, please note that all of the participants are muted to minimize background noise during the presentation. Also, during the webinar, we will be asking some poll questions. You can use the question window, we'll be joining our speaker to help facilitate those poll questions. I'll be jumping in and you'll have an opportunity to vote and see the results live. And we hope you enjoy participating in the polls. We think they're kind of a fun way to make the webinar a little more interactive for everyone. There will be a quick Q&A question at the end of the webinar, and you can use that little question window to ask your question at any time during the event. We will answer as many of your questions as we can during the Q&A. Any questions that we don't have time for, we will be able to answer by email. And also, following the presentation, you will receive an email with a link to the recorded webinar, so please keep an eye on your inbox for the notification that the recording is available. Okay, well, that's all for housekeeping notes, and I'm very pleased to welcome today's speaker, Dusty Vyas. Dusty is a senior scientist with Biosearch Technologies, and she specializes in plant genetics. And over her 30-year career, she's worked on the development of molecular markers at both James Hutton Institute and Biogemma UK, and she's been with Biosearch for the past 8 years, where she's enabled the translation of LGC's biotechnologies in order to for the global plant breeding community and species all across diverse samples types, including wheat and cocoa and rubber and rice and aquaculture as well. And we're very excited to have Dusty present today's topic, KASP Technology Accelerates Rice Improvement in South Asia. Go ahead, Dusty, take it away.
Great, thank you very much, Beth, for that lovely introduction. Before I go into the details of the project, I just need to say thank you to the groups that have been involved in this collaboration. This was an Agritech funded project and involved a number of partners, including APL, which is a private breeding company in Nepal. We've got NIBG, which is in Pakistan and is part of the public sector breeding. We have NARC as well in Nepal, which is also a public sector research institute. We have SCUAST, which is a university based in India. We couldn't have done this without the Agri Centre, who carried out the project management, got Bangor University, led by Catherine Steele and John Whitcomb, and without them, this project wouldn't have even started in the first place. So Bangor University took the lead in the feasibility project, and then LGC Biosearch Technologies took the lead in the industrial phase of the project. So, as we all know, rice is one of the most important global agronomic crop species, and rice is a staple food for more than half the world's population, making up more than 20% of calories for nearly 3.5 billion people. The majority of rice production is in Southeast Asia. And to give you an overall picture, as you can see on the graph on the left-hand side, the production of rice has increased from 450 million tonnes to nearly 515 million tonnes in 10 years. But as you can see on the graph on the right-hand side, the increase in production in Southeast Asia has not matched the demand in the past few years. And this is primarily due to floods and droughts that were prevalent in 2017, where we saw a reduction in production. So this is why it is very important to develop new traits and new varieties that are adapted to the climate changes that we are seeing at the moment and in the future. So when we look at the factors that influence the reduced yields and the development of varieties, two of the biggest diseases that cause these problems are BLB and blast disease resistance. Blast is a fungal pathogen which leads to losses of nearly 100%, and it's regarded to be one of the most serious diseases worldwide. It can be treated with chemicals, and up to now there have been nearly 20 R genes that have been cloned and identified, including PI9, PI24, PI25 and PI40. The graph in this slide shows the severity of blast disease across India, and it shows the variation according to the rainfall and season. BLB has been shown to reduce yields between 5 and 85% and accounts for nearly 40% of losses worldwide. And this particular disease cannot be treated in any way. So the only way to get resistance to this horrible disease is to integrate resistance genes for BLB. And up until now, there have been 6 R genes identified, 3 of which are XA5, XA13, and XA21. Resistant varieties are therefore a major goal for all rice breeders, and using a single gene approach and multiple genes, and this is called pyramiding. Marker-assisted backcrossing has been used for the past 20 years to incorporate resistance genes, and these have been released in all countries, but it has been through the application of a technology called SSRs and PCR markers. One of the other key important trait for breeders is aroma, and this actually increases the higher value of rice varieties. And this trait increases security for farmers and means that they can get a higher value for their harvest. The most common fragrance allele is BADH2.1. And in Asian rice, it has been identified as a trait-specific marker and is a common target for the majority of breeders. And it's these markers that have also been developed as KASP technology. And our partner in Nepal, APL, are currently working on a variety called Sinolus aganda for this specific trait. This variety has been crossed with other desirable varieties through the identification of polymorphic markers and is in current breeding programs in Nepal.
All right, and this will bring us to the first of our interactive poll questions today. The question for you is, are you currently using any molecular markers for variety development? And if yes, which marker detection chemistry are you using? Are you using microsatellites, SSRs, and or STSs, sequence tag sites? Maybe you're using single nucleotide polymorphisms or SNPs and or indels, or maybe none. Maybe you are not using molecular markers for variety development. So please let us know, choose your choice. If you have any additional comments or questions, please remember to use that question format there in the webinar screen. And we'll take a look at the results live. All right. And it looks like everyone's had an opportunity to vote, and more than half of the population here, more than half of our audience is using SNPs and or indels, Dusty.
Well, that's fantastic. That's very, very good news. So I'll carry on. So if we look at the rice genome as a whole, it's actually quite a tiny genome, and it's only about 430 megabase pairs long. It is a highly polymorphic genome. There are over 40,000 protein coding sequences. There's about 35% with repetitive sequences. And of these, there are 48,000 SSRs. When we compare two varieties, this will give us a true indication of the polymorphic nature of rice. So when we compare the two different varieties, over 4,500 polymorphic SSRs can be identified. There's about 39,000 indels, but there are nearly 338,000 SNPs. So this gives you an indication of the variability and the types of markers that could be targeted in rice in particular. So if we look at the project when it started back in 2014, there were over 18,000 SSRs. And at that time, we had 2,055 KASP markers available. And of course, we know from the previous slide that there are fewer SSRs available than SNPs in the rice genome. When we look at the safety of running SSRs in laboratories, we can see that there are concerns around the use of things like acrylamide for running high-resolution gels, or we have the problems associated with ethidium bromide that are run in agarose gels. And both of these chemistries and these chemicals are used for the application of SSRs. When we look at KASP, on the other hand, it uses a fluorescent detection system, which is simpler and much safer to use. Throughout, and the throughput for SSRs, of course, is very, very low. We're generally running things on gels. Whereas when we look at the KASP technology, this is an ultra high throughput system, and that's why this was chosen in particular for the targets in this project. For the feasibility project, we actually used the Japonica reference genome in the development. And out of the 2,055 GCP markers, only 1,091 were found to map to the indica reference genome. So as we see that KASP also has the benefit over arrays, they're not fixed, and they can be selected for specific crosses, and can reduce the number of, and then you can go on to reduce the number of assays in the advanced generation, which shows the flexibility of KASP. Once the KASP assays are actually set up, there's no additional cost, and I'll take you through the design process that was used in this project and available to all breeders. So looking at KASP technology in more detail, KASP chemistry is a PCR-based genotyping option which involves an amplification of specific regions of DNA using primers. As part of the KASP genotyping, the assay components include a primer mix, and this is typically 2 allele-specific forward primers and a single common reverse primer. As you can see in the slide, the KASP assays, the KASP primers are not actually labeled, which in itself brings down the cost of the production of the oligos for the primers in KASP. The allele-specific primers only differ at the 3' end. And in this example, we can see that the differentiation, the allele-specific base for one of the primers is a C, and the other allele-specific primer is an A. And during the PCR cycles, these primers are competitive. The reverse primer is common to both sequences and will bind to all DNA samples. Another component is the master mix, and this contains all the components for the PCR reactions, including enzymes, magnesium, DNTPs, and it also contains a universal FRET cassette, which does not generate any signal before the PCR reaction, as the fluorophores are quenched. Of course, you need the DNA template, which is extracted from the individual samples, and this enables the KASP technology to be extremely efficient and robust. So For this particular project, we used KASP by design, and in this scenario, a customer submits a design sequence. The target variation, the SNP that we're trying to target, is indicated by the square brackets. We require 50 base pairs on the flanking sequence on either side of the target. Any ambiguous bases can be submitted as IUPAC codes and will allow the incorporation of any wobble bases that we are seeing. All of this is done, all the KASP assay designs are done through our proprietary Kraken software. And as I said before, KASP assays include the development of 2 allele-specific primers and a single reverse primer.
Okay, and this will bring us to the second of our 3 audience polls today. And this one, again, asks about experiences in your lab. So in your laboratory, do you have any challenges in identifying and using molecular markers like SNPs and indels? So no challenges. We are already using SNPs and indels routinely. Maybe you need guidance or services to discover SNPs and indels, or perhaps breeding consultation to carry out association studies. Perhaps you need both, both the second and the third choice there. Or maybe you're completely comfortable with your current method for genotyping. It looked like from our previous poll that over half of our audience was currently using SNPs and indels. So we're curious to know what kind of challenges you may or may not have. We'll give you just a couple more seconds to vote here, and then we will all see the results live.
I want to know.
Right. Yeah. Oh, looks like some folks could use some guidance or consultation. About 33% of the audience, and then about a quarter of the audience is using both either or SNPs and indels routinely with no challenges. So
Great.
good to know. Thank you for taking a part in our poll.
Thank you, everyone. So the other technology that was used as part of the feasibility project was the conversion of known SSRs to KASP. And we did that through long-range PCRs, which covered the SSRs and would provide us enough sequencing information to identify potential SNPs that then could be converted to KASP technology. The service that we have provides NGS sequencing, data analysis and scoring, and we also design the assays for you, and then of course we can carry out the genotyping as an alternative to running SSRs. So this was one of the other technologies that was applied in the feasibility project or the RICE acceleration project. So the feasibility, the Catalyst project funding that we got from Innovate UK had the collaboration between NARC. We had novel rice lines that were developed through DFID funding. We were able to identify resistance genes and sequences from a vast database that from the ERI database, as well as the Nepalese government's, the Nepalese Institute's databases that they have there. All of this, the information and the collaborations were there to develop resistant lines specifically for BLAST and BLB. And all the work was carried out at NARC and Anilbu, which is the company in Nepal. And we wanted to take these resistant genes and incorporate them into new varieties. And I'll give you a bit of information on the development of that later in the project, in the presentation. So this is a picture of John Whitcomb, who's one of the leads in the Nepalese rice project, looking at rice development in Nepal. So we wanted to identify variation at a genome-wide level. We then wanted to design KASP assays for polymorphic markers. And this was with the intent of developing KASP genotyping for public sector breeding, and which in turn will develop varieties that are going to be grown by local farmers and hopefully improve their livelihoods and provide them with a degree of security for their harvesting. So, as I said, at the start of in 2014, we had publicly available 2015 KASP markers, which were developed through the Generation Challenge programme. But when we actually took these markers and compared them to the indica reference genome, we found that there was very low coverage. And on the graph on the right-hand side, you can see that there are a vague dispersion of markers throughout the genome. There are still areas where there were no markers actually available. Around from 284 and 414 markers were actually informative, and these were the markers that were used as in the pairwide crosses in the 9 lines that we used in this particular study. So what we did after carrying out resequencing of 9 Nepalese varieties, we then mapped that against the indica genome and compared the coverage, and this is what we got. So on the left is the coverage of markers that we have seen with the existing markers at the beginning of the project. But once we aligned the resequencing data from the 9 varieties in this project, we were able to increase the coverage to a point where there wasn't a single area of the genome that would not contain markers that could be converted to KASP. So, and it was almost a 100-fold increase. So using these markers and using varieties from Erie where there is blast and BLB resistance, so they were the donor varieties, and the recurrent parents on the left-hand side contained no resistance to blast or BLB. So it's from these crosses we were able to select through back crossing progeny that contained resistances to both diseases. Different combination of XA5, XA4, XA7, XA13 and XA21 were incorporated into popular rice commercial varieties. I mean, this was done using SSR and sequence tag approaches before, but our approach was to use a high throughput genotyping option using KASP, which would not only allow faster identification of potential progenies with potential disease resistances, but was able to carry out the genotyping before the next selection was taken in the field. When we look at more detail of the resequencing efforts that was carried out, and this was done through paired Illumina sequencing, we had a very high mapping rate. It was up to 93 or 94%. We had very good genome coverage, and the depths of reads that we got gave us a great deal of confidence in the data that was generated by our team in Berlin, our Berlin sequencing laboratories. So having all this data, then we had to develop a pipeline which could potentially identify good KASP markers. As we all know, all SNPs may not be conducive for the development of KASP assays. So the pipeline involved filtering through the evidence of the variant, which by which we mean that there is a polymorphic SNP at that position, the identification of indels in the flanking regions, the number of ambiguous bases within the 50 base pairs on either side of the target SNP, the presence and absence of repeat sequences surrounding the SNPs, and of course, one of the important things was to ensure we had a good read depth to identify absolute sequences. So the great news was that we've gone from, as you can see in this graph on the right-hand side, this shows the numbers of markers that were available when we did a pairwise comparison of the varieties. So the numbers on the right-hand side are in the hundreds, and this is using the sequences that were previously available. And then on the left-hand side, you can see the massive increase in availability of potential markers that could be converted into KASP in the 9 varieties that were used as part of the resequencing effort. And this is where we see an increase of around 100. So we've gone from having a few markers to having an incredibly large number of markers to select from. And all of this information with the pipeline for selection was applied to the data that was generated from the resequencing efforts. So the outcome overall, we were able to identify nearly 1.3 potential KASP assays on the 9 genomes. And this meant that we could identify potentially nearly 92,000 functional markers. The numbers of markers went from, I think it was 1 marker per KB to 3.1 marker per KB, which is massive and which is very important. Another outcome, we validated novel KASP assays in a range of crosses. We demonstrated that KASP conversion near SSRs is possible and can produce SNP markers which are just as good as the SSR, and of course increases the throughput and efficiency of the selection process. We had the collaboration between private and public breeding sector in Nepal, and all of this was published in a paper in 2018. So this was the first half of our journey together, should I say. So we've carried out the feasibility study, we've resequenced nine Nepalese varieties, we've compared them to the reference genome, and we have a pipeline which could develop robust KASP markers. So then we moved into the next stage, which was the industrial phase of the project. And the target for the industrial phase of the project was to increase the uptake of KASP chemistry across Asia and to improve rice breeding and ultimately improve the livelihood of millions of farmers in other rice programs. So as part of this project, we welcome NIBGI and SCUAST, who joined the project in the industrial phase. Some of the targets were also expanded, and we wanted to improve drought and abiotic stresses. We looked at brown spots and other significant diseases. And we aim to improve the efficiency of selection process and adaptation for these stresses and reduce the yield penalties through the selection using KASP. This will also reduce the pesticide usage and increase the local economic benefits for smallholder farmers. So in this map, we can show you where our partners are actually located. On the left, we have Nipchi, which is in Pakistan. We have Skuas, which is based in Kashmir. We have APL in Nepal, and NARC on the right-hand side, which is based in Kathmandu. We've now, at the end of the feasibility project, we began to do advanced trials in the different agronomic regions in Nepal. In yellow, we have the lowland variety development, and then we also have, at NARC, we have BLAST and BLB screening options. And this is to show you that, you know, we involved the Nepalese breeders all over Nepal and helped develop these new varieties. So going into more detail about the aims of the industrial project, it was to build on work carried out in the feasibility studies and make these KASP assays available to all rice breeders. We wanted to ensure that the KASP designs could be utilised in a wider range of rice varieties, and the other aim was to identify suitable SNPs in the indels to replace SSR markers and the confirmation of phenotypic trait-specific markers. We wanted to ensure that the validation process of the 4,000 markers that were identified in the feasibility study through our technology and through our service capabilities. And one final objective for the industrial phase project was to actually develop a database which could be accessible directly from LGC. And all of this I will go into a lot more detail momentarily. So to actually increase the relevance of the sequencing data and the relevance of the markers that were identified, we took the sequencing data from the feasibility study and we compared that to the RICE 3000 genome project, which was developed by ERI. We aligned the sequencing from the project with 119 different varieties, which included a range, at least one from every country. We incorporated different types of rice, including intermediate types, basmati and japonica. And in this example, we actually used the updated indica reference genome, which is R498. So as part of the process to improve the applicability of the KASP assay designs, we had to look at the submission sequences. So in this particular example, we've got we've got a pairwise comparison of two varieties. The SNP can be identified by the underscore, and it's represented by the IUPAC-S, which conforms to a GC SNP. We have variation in the surrounding flanking regions within 50 base pairs, which have also been replaced with a IUPAC code to incorporate variation. But if these markers were developed, they would work very well in these two particular varieties. But then if we then applied these markers to another variety, we can see that the KASP marker that has been developed would of course work with varieties A and B, but would work not so well with varieties C and D in this example. So to do that, by looking at the 3000 project, we were able to identify further variation in the sequence and consequently produce a KASP primer or a KASP assay which was applicable to a wider range of varieties that have originated from different countries. So we'll take a tiny break there for another question.
All right, thank you, Dusty. And this is the last of our 3 audience polls today. The question for you is, do you want to, or maybe you already are, using KASP genotyping in your breeding program? And if so, how many samples and markers do you usually run on a monthly basis? Do you run less than 100 or more than 100 samples? And then how many markers? Is that less than 100 or more than 100 markers on a monthly basis? There's a little bit of variation here, right? And then maybe you're comfortable with your current approach that does not use KASP. If that's the case, then you can go ahead and select the first one. And we look forward to seeing the results.
Is this going to be live? Are these live polls?
Yep, we'll see the results
Okay.
live in just a minute. Just to give our audience enough time to complete their choices. And it looks like more than 100 samples with less than 100 markers is the winner, although there is some variation. So we have a variety of different sample numbers and marker numbers that folks run on a monthly basis, Dusty.
Oh, I would expect that, actually. I thought there would be a range.
Looks like you were spot on. All right, we'll let you continue with your presentation. Thanks everyone for participating in our polls.
Yeah, thank you. Thanks a lot. So by taking into account the variation of the different genomes, the robustness of the KASP markers identified in this project will be of value to more breeders. And this will make these markers unique. I don't think anybody else has gone through this process of looking at large numbers of varieties to develop KASP assays. So the panel that we've developed, the 4,000 markers that we have developed, will be a unique set of markers. The designs were based on the comparison of nearly 119 lines from the 3,000 project, and there was 13 additional resequence line, including the ones that were developed in the feasibility project. Using this approach, we were able to identify nearly 1.2 million potential design markers. And as we can see on the right-hand side, the target alleles would cover nearly 90% of varieties that were tested. So another part of the project was to look at existing SSRs and whether we could use the resequencing data to identify potential alternative KASP markers. We did this by downloading nearly 20,000 SSR primer sequences. We blasted that against the Japonica and Indica genome, and we were able to identify additional 140 marker primer pairs, and these were from literature and publication. And using this approach, we were able to see that there were only 30 markers which were indica specific, and of these markers, only 2,200 were only specific to Japonica. So this gives you an idea of the numbers of markers that we are looking to develop alternative KASP markers for. So through the industrial phase project, we were able to identify nearly 5,000 markers which had been validated across 170 diverse varieties. And all of this information will be available through LGC's database, which is currently being developed and will be available through our website. Of the 4,867 SNPs identified, 3,365 were identified to be near SSRs. And the great news from the industrial phase project is that we have 33 aromatic advanced entries now under test in a multi-locational field trials. And for me, this is an immense achievement of the project. In NARC, they looked at the development of a blast disease resistance, and this particular slide shows you the improvements in rice varieties, not only that have blast disease resistance, but were also cold tolerant. And all of this development was done through the Nepalese Agricultural Research Council, and it is another aspect that we can be very, very proud of. In NIBGI, they have been using 215 KASP markers, and at the moment, association mapping is being done for yield and yield-related traits. We have a differentiation between Basmati and non-Basmati lines are also being genotyped with KASP markers. And overall, the KASP markers are being used in the Rice Pakistan breeding program for the incorporation of BLB resistance in Basmati rice, along with other grain quality traits. This has been huge for SCUAS, where they had deliberations held on KASP genotyping to see the potential on how much further the application of KASP can be carried out in Indian rice breeding programs. And here is an example of where we've got scientists and scholars actually carrying out leaf sampling, and that was done using our leaf sampling kit, which is a box basically that you can take to the field. You can directly sample the leaf material, apply the desiccant, send it to us. We can do the extraction, genotyping, and just provide the data. And this shows it being done in situ in glass houses, which were developed as part of the Nepalese rice project. We were further honoured by the Director General of the Indian Council of Agricultural Research, and he was actually briefed on the development of KASP markers in this project, and he apparently was very impressed and is encouraging of the development of new technologies in India. So I think I've come to the end of this little webinar. I hope you found it useful. But before I go on, I need to put out thanks to Innovate UK and DFID funding for this particular project. We need to thank Dominique Favan, who was one of our product managers. I'd like to thank Rian William for her support. This could not have been done without Catherine Steele or John Whitcomb, and Mark, who's our bioinformatician, who was absolutely brilliant. Dr. Mohammed Arif in NIBGI. We've got Resham Amji, Ram Baran, who are both based in Nepal. We have Rajiv Dakal, Anju Bandi, Dipender Ira, and Maheshwara, who are at APL in Nepal. And Freddie Reed, who's no longer part of the project, who was a brilliant project manager, and he was based at the Agri Epicenter. So I hope you've enjoyed that, and I'd just like to say thank you to everybody.
All right, thank you, Dusty, for the presentation. And now we do have some audience questions that we can go through here and start to answer those. The first one is, I am already using KASP assays for genotyping. How can we improve the efficiency in a single setup laboratory like ours?
So to increase if you've got a single setup laboratory, which you're actually using KASP at the moment, and you want to increase your efficiency, depending where the bottlenecks are. Is it in the extraction part, or is it in the genotyping part? So if we want to increase efficiency in the genotyping aspect of your laboratory, then we can look at alternative instrumentation that could be put in your laboratory, such as the meridians. We have hydrocyclers, which can improve the throughput. So there are instruments and options within our SNP line offerings which could improve your efficiencies. I hope that answers the question.
Great, thank you, Dusty. And then the second question that we have is, what differentiates the new KASP product from the previous run? This must be a current customer who has gotten some notification that we are updating the formulation. Do you want to address that one?
Yeah, I can do that. So the old formulation contains a chemical called Triton X, and the new formulation is actually Triton free, and this is because there have been EU regulations through the REACH initiative which stipulated that Triton could not actually be used in the formulation. So we've gone through development, validation and communication that the KASP formulation has now changed and is Triton free.
Great,
and there shouldn't be a difference.
And those new formulations will be commercially available starting in January when the REACH regulations take effect. So right now the legacy formulations are available until the end of December, and then we'll be making the switch. Okay, we have a couple more audience questions here. I have used KASP in oats. Now I am working in mulberry. The reference genome is available with 22 genotypes. Resequencing is available. Is it feasible to use 25,000 SNPs with 700 genotypes?
Using KASP technology, I would say it is absolutely possible, but I'm not sure it would be the most cost-efficient approach. There are alternative technologies that we have, such as SeqSNP, which probably would be more applicable in this situation.
All right, and if the audience has any questions about SeqSNP, they can type those questions in, and we can address them either today or address them by email afterwards as well.
Absolutely.
Right.
Absolutely.
Another question: how to convert, or how would someone convert SSR to SNP?
So our current service offering is the fact that we need a reference genome. We need to know the locations of the SSRs, and we would obviously need, and this is the SSR conversion is only applicable to trait-specific SSRs because you only have a single allele which is associated. SSRs also have multi-allelic options where we're looking at diversity studies where the numbers of SSRs may vary according to the variety. The SSR conversion is not actually possible for those ones, but for trait-specific SSRs, it's possible. So we would need a reference genome, we'd need positive and negative samples for your trait that you're targeting. We would need a minimum of 48 samples, and of course, the reference genome would be absolutely imperative in the SSR conversion service.
Great, thank you, Dusty. Okay, and another question about this particular project. Did you use DART in this project, so D-A-R-T?
No, we didn't. We used whole genome sequencing, which was carried out by our Berlin laboratory, which is our sequencing, well, one of two. We have one in Middleton, and the guys in Berlin also did sequencing. So no, DART was not used for this. We used whole genome sequencing, which of course provides you with a lot more detail and a better understanding of the genome.
Okay, thank you, Dusty. Here is one. Are there any KASP assays developed for any tree species?
I do know of in pine, I think there are some KASP markers that have been developed. I mean, what I do, which is a little trick for everybody, is to put your species, KASP and markers, and generally you can pull up all the publications that are available. And if there are panels that are available publicly, that's a good way to do it, or drop me a line and I can see what I can find for you.
Do you use just general Google for that search, or do you use Google
Yeah.
Scholar?
No, I just use Google.
Just use Google. Okay. All right. Next question. I had SSR markers that I wanted to test for linkage to a particular trait. I was utilizing ABI capillary electrophoresis. Can I convert those panels to KASP?
If it's trait specific, yes, that is feasible.
Okay. All right. Right now we are in the process of to KASP assay genotyping development for oil palm. I wonder if you have any KASP genotyping databases or also application for oil palm.
We do work with oil palm, and KASP does work very, very well with oil palm. We have major companies that we are working with. So yes, it can be used with oil palm. Do we have anything that's publicly available? Not off the top of my head, but we can have a dig around.
Mm-hmm. We have a couple of customer case studies, I believe, around palm
Yeah.
oil.
Oh yeah, we
So
do. We do.
that we might be able to share via email as well.
Cool.
When designing KASP assay primers in polyploids, is it more desirable to have subgenome homology with the SNP or with the SNP present in only one subgenome?
So we have an option which we didn't talk about in this particular webinar, was the fact that we can actually anchor our KASP assays to particular subgenomes. So having information on the homology to a specific subgenome is extremely useful. But through our design process, which is not necessarily automatic, and it would involve developing the markers as a KOD and a manual design, but we can make those in polyploid species as long as there is a small amount of variation within the subgenome. We will attempt to anchor the KASP primers to that particular subgenome. So we have used it quite extensively in wheat, which obviously everybody knows that we have the ABD genomes, which have a degree of similarity, should we say. But within our capabilities, yes, we can anchor to specific subgenome in polyploid species.
Okay. And you mentioned KODs. It may be worth just reminding our audience what the difference is between a KBD and a KOD.
So KBDs are basically submission of your SNP sequence, which can include variability in the flanking regions using the IUPAC codes, as I explained in the presentation. And they will go through our Kraken proprietary design software. And all it will do is take those sequences, design the KASP assays, and send you those assays as a design-only option. But with KODs, we go through a full validation. We would need samples, a minimum of 22 samples. We go through a wet lab validation, which means that we will run the KASP markers on your samples to ensure they are optimized for your particular target. And this may involve some redesign. So whatever, generally, we will go through to the nth degree to try and get these KASP markers working for you. And for me, when you're looking at something like trait-specific markers, which are going to be used for a long, long time, and in your breeding programs over a long period of time, if you want to ensure the viability and robustness of those markers, the KOD option is always the best bet. We will validate those markers for you.
Great. Thank you, Dusty. Similar question to one we had above, but do you have any genotyping panel for cotton?
Not on our website, but I do know of papers where they are talking about KASP markers for cotton. So please get in contact and I will try and pull them out for you.
Okay, thank you. And Dusty's email address is right there on the screen if you want to contact her. We'll send all the follow-ups to Dusty.
Well, no, actually, we should actually put tech support, use the tech support email in there.
We can, yeah, we'll send that information out in the email follow-up to the webinar.
Okay, yeah. So if I'm not around there, we've got a team that can help.
Sure, absolutely. And then another question, could you give a brief explanation about KASP assay development workflow? For example, development of KASP genotyping for oil palm. So what does the overall workflow kind of look like?
So, I mean, so if you have a sequence which has been identified in publications or databases or through resequencing efforts, SNP discovery, it might have come from an array. We need that, the SNP sequence, as we've talked about. We would design, as a KBD, we would design those markers and send them, either use them in a service project or send them to you. But the first step, which I really want everybody to understand, is that in a breeding program, when you're going to be using these markers as part of the process flow, you need to use this on your parental varieties to ensure that these particular markers are present. You know, and then once you've confirmed this and the KASP marker is polymorphic between two parents, then you can go on to use that at various stages in your breeding program. So you do your design, we produce the KASP assay, we send it to you, you check it on your parents, identify that is polymorphic, and then you can go on to do the extractions continually throughout your breeding program. But that validation in your parental varieties is absolutely key.
Makes sense. Okay. Here's another question. Do you have experience in KASP development in fish species in aquaculture?
Hell, yes, of course we do. Yeah, and we also have now, we have the BioArt fish kits as well, which means that the sampling is not just for plant species. And yes, for aquaculture, we do a lot of aquaculture genotyping using KASP, and people are now using our BioArt fish kits to collect fish fin samples which are stabilized, and they're sent to us, and we can do the extraction and KASP genotyping.
Yep, so that's the genotyping services option. And then we can also do the assay design for anyone who wants to have the assay delivered to them for use in their own lab as well, right? So a couple of different options for you. Yeah.
It's whatever what people want to do. Do they want to do it themselves? Great, that's up to you. But if you want us to do it for you, we can do it for you as well.
All right, and then another question, we have time for one more here, just to bring it all back and tie into this particular presentation. Will the varieties developed through this project, the rice varieties, be adopted by local farmers in Nepal and Pakistan?
There's already interest, so as far as I know, yes, absolutely, that's why we're doing it. We want to help people, and we want to ensure farmers in Nepal can grow varieties which have not only the disease resistance which we were targeting in the feasibility studies, but we can help improve yields, adaptations to changes in a climate in the next few years, looking at aroma, looking at yield quality. So with the 4,000 markers, there is a range of targets that can be used. And one thing I didn't mention in all of this is that those 4,000 markers could potentially be used as the genomic selection approach. because of the coverage that it provides across the whole genome.
Great. Okay. Well, thank you, Dusty, very much. That's about all the time we have for today. Thank you to our speaker, Dusty Vyas. Thank you to all of you in our audience today for sticking with us, for your great questions, for participating in our polling. We hope you enjoyed the webinar. And as a reminder, we will be sending a link to the webinar recording within a few days, so please keep an eye out for that email notification. Thanks again to everyone for joining us, and we'll go ahead and close out the webinar now.
Thank you, everyone. Thank you.